Hello, I'm back. Okay, so I'm going to do this real quick. This is going to be just a quick review of the steps of meiosis. I'm also going to show you one with the beads if you need that visualization. The worksheet you're going to do on this was meant to be done with the beads. What you're going to do instead is just draw this process out. Okay, pretend like you have the beads or whatever, but take the time to look in your book, read over meiosis, because basically we have one other lab to do, which I'm going to wait and assign you until after spring break. And then you're going to have your test. So you will have your test the week after spring break, whether we're in or out of school. This will be your last lecture day. And then we'll have a lab the week after spring break, which I'll probably post during spring break. And then you'll take your test. All right, how do we make sperm and egg cells? Well, that's called meiosis, my friends. Now, don't get stuck on the humans. Yes, in our meiosis, we reduce the 46 chromosomes to 23. But is that the case for the cat? No. Is that the case for whatever organism we're dealing with other than humans? No. You take the diploid number and then you have it, but you guys get kind of stuck on, oh wait, I'm gonna make that bigger. Sorry. You get a little bit stuck on the human count of 46, and then you start to kind of struggle when we start to talk about other creatures, okay? Looks like this cat's gonna make a little visual here. Oh dear, it's the laser. Okay, so here is meiosis, and what we're doing is we're having the chromosome number to 23, and then what's the point of fertilization? To restore the chromosome number. So the egg and the sperm come together and boom, we have 46 again. Okay, so that happens for all species, but they don't keep the number 46. All right, so this is a chart for you to fill out. What do you remember about meiosis versus mitosis? There are a ton of test questions that really make you differentiate between mitosis and meiosis. Mitosis, who does it? Everyone does. What's the advantage? You get an exact copy of the parent cell. The daughter cell is identical to the parent cell in mitosis. Meiosis, who does it? Egg and sperm cells or gametes. And what would be the advantage? Well, you get genetic variation because the daughter cells are genetically different than the parent cells. And that happens because of crossover and just independent assortment. We will talk about all those later. Okay, meiosis is the production of gametes. All species have this alternating stages of all eukaryotes, where you have the diploid stage, which for humans is the longest stage, right? That's what we're most familiar with. The egg and the sperm stage is the haploid stage. That is the stage that we're not as familiar with. Some plants live predominantly in the haploid stage, and they're only in the diploid stage for a short time. But the biggest thing and that's what you're going to see in our lab dealing with the sporophyte lab that we'll do, is their predominant life cycle is going to be in the haploid phase. All right. Fertilization restores the chromosome number. That's what we do. That's why we do the fertilization thing. All right. Meiosis must reduce the chromosome number by half. Why? This was a slide from General Bio last year. Because this is a preservation of species. This just shows you if you have mom and you have dad, and if they did not have their chromosome number, oh my Lord, we have a child with 4 chromosomes instead of 2 each, and every generation it would get bigger and bigger and it'd be terrible. However, if we have the chromosome number, then Upon fertilization, we restore the chromosome number, and that is the point of gametes, is that we can maintain the chromosome number from one species to the next. All right, homologous chromosomes play a big role in meiosis. The point of meiosis is to separate these, right? You've got 2 copies of every gene. Your mom, let's say chromosome 1. This isn't true, but you've got the eye color on it. Your mom gives you a gene for eye color. Your dad gives you a gene for eye color. The goal of meiosis is to separate the homologous chromosomes so that the sperm or the egg cell only gets 1 of the genes for eye color. You don't need 2 because you're going to find a mate and they're going to have their version. Okay? But it can't be random, because then suddenly we got to make sure that every sperm or egg gets 1 copy of each of the homologous chromosomes, so that you're not walking around with a sperm or egg without any eyes or something. So homologous means the same information. Is it identical? Not unless you mated with your identical twin, which wouldn't work. But they code for the same genes. All right. Here is comparing mitosis and meiosis. This is a very good demonstration. If you look here, metaphase in mitosis, the chromosomes line up in a single file. In meiosis, metaphase 1, do you see where they're double filed? The homologous chromosomes line up in a double file line. After telophase 1 of meiosis, the daughter cells are haploid. That's right here. Look at this. We've now separated and the daughter cells are becoming haploid, but they still have their sister chromatid. The whole point down here of meiosis 2 is to separate the sister chromatid. But it is after telophase 1 that we have halved the chromosome number. And I will show you this with the beads on its own video. DNA replication during the S phase is where we make the sister chromatid. Now we're going back to the cell cycle. The first division of meiosis, do you see this in bold and underline? You have to know this to get through this test. The first division of meiosis separates the homologous pairs. So at that point, we have halved the chromosome number. The second division has the sister chromatids. So these two, they're like, oh, I love you so much. We're soulmates. No, we ripped the homologous chromosomes apart. Now the sister chromatids are like, after meiosis 1, well, at least I still have you, sister. No, we ripped the sister chromatids apart. And now we have 1 copy of each chromosome without a sister chromatid. And yes, you end up with 4 gamete cells at the end of meiosis. There is a tricky question, like you have to know the difference on the test of mitosis and meiosis. And you really need to understand that the homologous chromosomes are separated during anaphase, meiosis, anaphase 1, or telophase, I guess you could say anaphase is when the homologous separate. And during mitosis, it is during anaphase that we separate the sister chromatids. When you read the test, people often make mistakes because they're not looking at the very simple things of what happens in the PMAT cycle, prophase, metaphase, anaphase, telophase. You get so caught up in what's the difference between mitosis and meiosis, you forget that if it says something like during metaphase 1 of meiosis, homologous chromosomes are separated. No, that's not what happens during metaphase. Homologous chromosomes line up. Okay, so just make sure that you read everything. All right, this is just showing you where, yes, I've said this so many times, homologous chromosomes separate in meiosis 1, meiosis 2, it's the sister chromatids that separate. Now, there is a mistake here. This should be haploid. So please note that this right here should say haploid because they don't have their sister chromatid. The biggest difference here, I know I'm going through this fast, but you should know this from last year. Please read through it and text me with any questions. But prophase 1 is when the crossing over occurs. When they pair up, that's where we have the synapse occur, okay? Crossing over, this is the definition that's on your test. Specific proteins break two strands of non-sister chromatids. Yeah, these guys are non-sister chromatids, and they rejoin them, okay? Once they form together, we say it's a synapse, but the chasm is the point where the crossover occurs. You don't really need to know the difference between those words. You need to know the synapse is what they form when they come together. Okay. Again, this is in green and bold. Probably need to know that. Meiosis 2 produces gametes, one copy of each chromosome, and thus one copy of each gene. Please go through your notes on this and look at anything that looks different or new to you. Ask questions, but I'm not going to lecture through all this. That's why you're going to do the worksheet. Which of the following would happen at the conclusion of meiosis 1? That's a test question. And at the conclusion of meiosis 1, basically those homologous chromosomes are separated. When are chromatids separated from each other? The sister chromatids, that's meiosis 2. All right. At the end of meiosis 2, how many cells will there be? There'll be 4 cells with 20 chromosomes in each. When does crossover occur? Prophase 1. All right, that's it for now. And I'll just give you a quick shot of Unicorn Girl. Unicorn Girl. Oh, no. Unicorn Girl.
Unicorn Girl. Unicorn Girl.
Say goodbye. Oh, say bye.